Hello, my name is Andy. In this video, I'm going to explain why I think that you should vote for me for the 2016 Bioanalysis Young Investigators Award. All the great videos on the internet involve cats, and I thought I'd make this one no different. So I want to first define what I think of as bioanalysis. We want to understand how organisms work. To do this, we need to understand how individual component cells work and how individual molecules within those cells conspire through their many interactions to give rise to the emergent phenomena that we see in organisms. Cells are hugely complicated environments comprising hundreds of thousands of types of molecules, and so bioanalysis is concerned with understanding how all of these interrelate. Because these mixtures are so complex, there's a continuous need for improved methodology to drive our understanding forward and show us exactly what happens inside a cell and inside organisms. I'm particularly interested in proteins. These molecules are simple organic polymers from the perspective of an organic chemist, but they fold up and adopt a myriad of functional conformations. From a theoretical point of view, it remains highly challenging to predict exactly which state a protein chain will adopt under a certain set of conditions, and so there remains a great need for experiments to tell us which form is present and how things interact. Many methods focus on the natively structured forms of proteins and require highly purified samples, but these don't give us the complete picture. In my career, I've been drawn to understand other commonly adopted forms of protein, and to develop methods that still work even when we have highly combinated mixtures and dynamic proteins, as we do in the interior of the cell. Studying these non-traditional states is an exciting opportunity for bioanalysis. To make progress, I've focused on three cutting-edge techniques: nuclear magnetic resonance spectroscopy, ion mobility mass spectrometry, and electron microscopy. NMR provides information about individual atoms, whereas IMMS and EM give information about the global shape, size, and oligomer distribution present. I've developed new methods for each, finding that the detailed physics give each a particular set of strengths and weaknesses, and together they're both powerful and highly complementary. I've developed novel computational methods to improve our ability to extract information from the raw data for each of these, and to harness them together in structure calculations. To illustrate what this lets you do, I've recently applied it to give us atomic resolution structures of individual oligomers, where the system in question forms a heterogeneous mixture. This shows that we can get away from the need to isolate and separate mixtures and still get atomic resolution information and structures. A method is only as good as what you can learn by applying it. In the final few slides, I want to illustrate new biology we've learned through these bioanalytical methods. First of all, amyloid fibrils. These are associated with neurodegenerative conditions such as Alzheimer's disease. These are commonly thought to be formed irreversibly in tissues. My colleagues and I studied amyloids formed from a range of proteins and determined that there's always free monomer left over at the end of an aggregation reaction, showing that the process is reversible. What was most interesting was that at biologically relevant concentrations, the most stable form of the protein is amyloid, and not the functional form. This means that proteins are frequently metastable, and that amyloid formation and associated diseases are thermodynamically inevitable, hitting us when we're old. In another application, we've studied the cellular compartments known as membraneless organelles. These include bodies such as nucleoli and stress granules that are ubiquitous to eukaryotic cells, and very challenging to purify and study. By looking at a protein that forms one of these bodies, we've determined that their formation is driven by a simple polymer phase separation of disordered proteins. This finding shows that these bodies are effectively regions of organic solvent floating around inside cells. We have since shown that when double-stranded DNA goes into one of these bodies, it is substantially melted, and there exists a complex set of rules of trafficking that allow proteins to go in and out of these. These new results show that cells harness organic solvents as part of their natural life cycle. Moving forward, current work in my lab is focused on pushing these hybrid bioanalytical methods further to study how amyloid formation occurs in the presence of living cells and tissues, and how they are affected by its presence. This will allow us to understand how the cell's defences work to prevent amyloids, and will hopefully allow us to develop new diagnostic tools and therapeutic agents. And so in closing, I would like to thank you for watching this video. The bioanalytical research conducted by my group and I will be boosted significantly by your vote